Hello friends, myself BP Shah, PGT Biology, Kendriya Vidyalaya Keshapuram. Today I am continuing my previous topic photosynthesis. I have already dealt in last lecture that what is photosynthesis, what are the reactions involved in photosynthesis. There are various type of reaction, light reaction, non-cyclic phosphorylation, cyclic phosphorylation, C3 cycle, and the rest thing, that is in dark reaction we have already discussed about it. The carbon dioxide is going to be fixed and it changes into carbohydrate, and the assimilative power which are stored in the form of ATP as well as NADPH2 are used here. Use of ATP and NADPH made by light reaction used in carbon difixation. I have already discussed, this is called biosynthetic phase, dark reaction or Kelvin cycle. It is also called Benson and Bessam cycle, where it can be traced the entire cycle with the help of carbon 14 technique, with the help of technique known as chromatography and autoradiography. It occurs in stroma, in the chloroplast. Now, there are two main pathway of the dark reaction, that is Kelvin cycle, which takes place in C3 plant, because the first stable product is PGA, phosphoglyceric acid. So, this cycle is called C3 plant. The another most important which we have to discuss today, that is C4 plant. It is also called C4 cycle. There is no taxonomic importance of this pathway, because in a particular species or in a particular genus both type of cycle are found, like Euphorbia. Euphorbia is a genus. In species Corollata, the pathway is C3, and in Euphorbia another species Maculata, the pathway is C4. There is another cases like Allotropis semileata, where C3 and C4 both are present. So, this is the taxonomic importance, because in a particular species both cycle may be present or may not. Until mid 60, it was universally accepted that only C3 cycle is the only pathway to fix the carbon dioxide into carbohydrate, that is glucose, by C3 cycle. But in 1957, the name of scientist Co2, he reported that instead of carbon 3 compound, that is PGA, the first stable product C4 compound is found while working with dark reaction. The another scientist In 1960, Karpilov LOB, while working with the maize plant, he surprised that the first stable product is not phosphoglyceric acid, instead of that oxaloacetic acid is formed. This is not C3, this is C4 compound. This was finally proved that by Hatch and Slack. In 1966 in Austria, Hatch and Slack, while working with the maize or sugarcane, he confirmed that the first stable product is C4 compound, that is oxaloacetic acid. So, after 1966, it was confirmed that not only C3 compound is formed in dark reaction, C4 compound is also formed. It was already proved by the 3 scientists and finally by Hatch and Slack. So, this cycle is known as C4 cycle, no doubt, because first stable product is carbon 4 compound oxaloacetic acid. It was also known as Hatch and Slack cycle. Hatch and Slack found that there is a specialization in this C4 cycle plant leaf. He shows that the plant which shows C4 cycle, there is specialization in leaf structure. Normally, the leaf in dicot, the upper epidermis, there is just below the upper epidermis, there is parenchyma, that is called palisade parenchyma. Then vascular bundles are there, where xylem and phloems are there. Here the phloems are there, and there is loosely connected tissue that is called spongy parenchyma, and finally, there is lower epidermis, where most of the stomata are there. This one is called mesophyll, this one is called upper epidermis, this one lower epidermis. This one xylem, phloem, In palisade parenchyma, large number of chloroplasts are there, and photosynthesis takes place in the chloroplast. Light reaction takes place in grana and dark reaction takes place in the thylakoid. This is the normal C3 plant leaf anatomy. Now, Hatch and Slack found that in C4 plant the anatomy of leaf has certain specialization. This type of anatomy of leaf is called Cringe anatomy. Here is the structure. This one is the same as C3 plant. Here is the upper epidermis. There is no palisade parenchyma as you see, and the vascular bundle. Here is the vascular bundle. This one is xylem, this one is phloem, this one is lower epidermis. Here is a specialized tissues, specialized bundle which cover the vascular bundle, that is called bundle sheath. Or this one mesophyll. So, there is certain specialization in C3 leaf and C4 leaf. This anatomy is called Cringe anatomy. Here the difference is mesophyll have only one type of, and the vascular bundle is covered by bundle sheath. This shows the C4 plant leaf, which shows Cringe anatomy, which means German meaning that is hollow or width, which means curled or twisted. Okay. So, this is the specialization type of leaf anatomy in C4 plant. That anatomy is called Cringe anatomy, where the first stable product is formed, that is called oxaloacetic acid. In C4 plant there is a differences between the mesophyll tissues and the bundle sheath tissues. The anatomy of leaf of C4 plant is different from the leaf of C3 plant. In the leaves of such plant like C4 plant, we have seen that here is no palisade tissues is absent. So, first point is palisade tissues is absent. Bundle sheath around the vascular bundle. The chloroplast of the bundle sheath cell are large and without or less developed grana. It contains starch granules. In mesophyll cell, the chloroplasts are normal type, smaller but well developed grana. Here the basic difference is the mesophyll cell have chloroplast normal type, small but well developed granas are there. In mesophyll cell, C4 cycle is performed, and in bundle sheath C3 cycle is performed. So, in mesophyll cell mainly C4 cycle and in bundle sheath cell mainly C3 cycle is performed. So, in Cringe anatomy leaf both C4 cycle and C3 cycle is performed, but the first stable product is oxaloacetic acid. So, this is called C4 cycle or Hatch and Slack cycle. Now, what happened in the what is the mechanism or physiological process in C4 cycle? This one is the C3 phase. You just remind it, the carbon dioxide from the atmosphere, it is directly enters and it is accepted by RUBP and form first stable product, that is 3 phosphoglyceric acid, and second one, that is glycolytic reversal, where the glucose molecule is formed, and regeneration of RUBP, that is the third phase. So, in C3 cycle 3 main process is involved, that is carboxylation, reduction and regeneration. Now, what happened in the C4 cycle? In C4 cycle both mesophyll cell and bundle sheath cell are involved. Here the RUBP have no role. Instead of RUBP, the first acceptor of carbon dioxide in C3 cycle, here instead of that there is phosphoglycinol pyruvate is the acceptor of carbon dioxide. This phosphoenol pyruvate is formed in the C4 plant. In the process of respiration, we have discussed it later on, the first product of the glycolysis, that is pyruvic acid. This pyruvic acid are abundant in cytoplasm. This is the structure of pyruvic acid. Which are already present in cytoplasm. It is for the conversion of glucose molecule into the pyruvic acid. It is also called EMP pathway. It combined with ATP plus H3PO4, then it converted into phosphoenol pyruvate. Here is the double bond from the H3PO4, this phosphate came and COOH. This is PEP. It is also called phosphoenol pyruvate, and the ATP is breakdown, it forms AMP plus H2O. So, from the pyruvic acid the PEP is formed. This PEP is the acceptor of carbon dioxide in C4 cycle. So, we have already learned how this PEP is formed, and it is the first acceptor of carbon dioxide in C4 plant. Now, what happened in C4 cycle? PEP is formed from the pyruvic acid. Now, two tissues are involved, we have already seen in anatomy. Here is the mesophyll tissue, mesophyll cell, here is the bundle sheath cell. Now, in C4 cycle what happened in mesophyll cell and bundle sheath cell. So, the freshly formed this one is PEP, phosphoenol pyruvate, and this PEP receive the carbon dioxide and combine with carbon dioxide, it form oxaloacetic acid OAA. This is carbon 4 compound. So, this cycle is known as C4 cycle. Here the structure of OAA, COH, CH2, C double bond O, COOH. This is oxaloacetic acid. The first stable compound is oxaloacetic acid. So, the cycle is called C4 cycle. Now, this occurs in the mesophyll cell. Here is the two options are there. In mesophyll cell, it form malic acid and aspartic acid. As it depends upon the presence of enzyme as well as NADPH2 and NH3. Aspartic acid is formed when there is the presence of ammonia and NADPH2. Here the enzyme is involved, that is called transaminase. And malic acid is also formed when ammonia is not available, only NADPH2 are there, and the enzyme malic dehydrogenase. So, the oxaloacetic acid has two options. It depends upon the availability of the enzyme as well as the substrate like ammonia and coenzyme NADPH2. If ammonia and NADPH2 is formed, then it forms aspartic acid. If ammonia absent, malic acid dehydrogenase is present, then it forms malic acid. Now, both malic acid or aspartic acid form. Now, it move to the bundle sheath cell from the mesophyll cell. Now, this malic acid now available in bundle sheath cell. This malic acid now again form it pyruvic acid. And in these cases NADPH2 is released. The NADPH is used and it converted into NADPH2 because the malic acid release 2 hydrogen atom and the process involved with the help of malic enzyme, and it form pyruvic acid. During this process NADPH2 formation, here is the release of carbon dioxide also. This carbon dioxide is accepted by RUBP and it form PGA, 2 PGA, 2 phosphoglycic acid. So, you find here in bundle sheath cell the CO2 is released by malic acid, it is accepted by RUBP. So, C3 cycle is going to be operated and this pyruvic acid again return back to the phosphoenol pyruvate as we have already discussed here. This pyruvic acid now which is formed inside the bundle sheath cell. It is converted into PEP with the help of enzyme here kinase, and it also requires ATP and H3PO4. So, in another word we can say that the both bundle sheath cell and mesophyll cell are involved in C4 cycle, and the enzyme here when PEP converted into oxaloacetic acid, that is called PEP carboxylase. So, the C4 cycle involved both mesophyll cell and bundle sheath cell, and the first stable product is oxaloacetic acid. So, this one is called C4 cycle. So, now we are easily to say that there are two type of biosynthetic reaction, that is C3 cycle and C4 cycle, which is operated in different types of plant, or it also depends upon the region where the plant are present in the tropical region or in the temperate region. Okay. So, now the differences is conversion of phosphoenol pyruvate, it requires much of the energy. It is the loss of the plant, it requires 2 extra ATP to regenerate ATP and AMP. So, in total 12 additional ATP is needed for C4 cycle. So, the total ATP required for the C4 cycle is 30 ATP and 12 NADPH2 to synthesize one molecule of glucose molecule, which is much higher in comparison to the C3 cycle. But there are certain advantages. The C4 plant can absorb CO2 even from very low concentration, which not availed by the C3 plant. So, these are the basic differences between the C3. Now, here 12 additional ATP are needed in C4 cycle. It is very important for examination point of view. Now, C4 the total number of ATP, that is 30 and 12 NADPH2 molecules synthesize one molecule of the glucose. Now, what are the difference between the C3 cycle and C4 cycle? So, now we can easily show that here the CO2 acceptor in case of C3 plant and C4 plant, the CO2 acceptor is RUBP ribulose 1/5 bisphosphate, which is 5 carbon compound, but in case of C4 it is PEP phosphoenol pyruvate, which is 3 carbon compound. So, this one is the basic difference between C3 and C4 plant in acceptance of the carbon dioxide. Here enzyme is involved Rubisco, which is most abundant enzyme on the earth. Here the enzyme involved PEP carboxylase. So, in term of enzyme we can say that the here Rubiscos are there, here two enzymes are involved, here PEP carboxylase and RUBP carboxylase. Why two enzyme is involved? Because in C4 plant we have already seen C3 cycle is operated in the bundle sheath cell and C4 cycle is operated. When C4 cycle is operated in mesophyll cell, the enzyme required, that is PEP carboxylase. When the carbon dioxide which is released from the in bundle sheath cell, which is accepted for the C3 cycle, that is RUBP carboxylase. So, both the enzymes are involved in C4 plant, but only one enzyme, that is the Rubisco, involved in C3 plant. Most important point, the product. Here the product, first stable product is PGA, phosphoglyceric acid, we have already discussed. It is carbon 3 compound. Here the first stable product oxaloacetic acid, which is 4 carbon compound. So, on that basis carbon 3 and carbon 4, the name of cycle or the name of plant, this one is the C3 plant, this one is the C4 plant, this cycle is called C3 cycle and this cycle is called C4 cycle. Okay. As we have seen in Cringe Anatomy, the chloroplast structure. Here the chloroplast is one type, but in case of C4 plant, the chloroplasts are dimorphic in nature. Dimorphic nature, that shows the Cringe Anatomy, because here the vascular bundle is covered by bundle sheath cell. Now, in term of CO2 fixation, it occurs only once only. It occurs twice, because we have seen the CO2 is formed in bundle sheath cell, which is received by RUBP, and CO2 in mesophyll cell, it is received by PEP. So, these are the 5 main points which give the differences between the C3 and C4 plant. In term of region, the C3 cycle it is operated in temperate region, and C4 cycle it is operated in tropical region. In term of region. Now, the last one, light intensity. It occurs, both require light reaction, because in light reaction ATP and NADPH2 is formed, which is called assimilatory power, which are used in the fixation of carbon dioxide. But in C3 cycle, high light intensity required for few hours. So, this type of light intensity are present in temperate region, but this high light intensity in tropical region, most of the part of the day high light intensity is found most of the part of day. So, these are the differences between C3 and C4 cycle. Okay. So, now this is the pictorial view of entire. Here is the pyruvic acid, ATP converted into PEP, CO2 combined with the PEP, oxaloacetic acid, as we have already discussed. The oxaloacetic acid converted into malic acid in presence of NADPH2 and malic dehydrogenase, and when ammonia is present, is converted into aspartic acid. This oxaloacetic acid and malic acid and other acid, it is transported to the cell of bundle sheath cell. This entire thing transported to the bundle sheath cell. Malic acid, it converted into pyruvic acid, and CO2 is formed. It is accepted by RUBP and PGA is formed, and sucrose is formed, and pyruvic acid again for PEP for the next reaction of carbon dioxide fixation in C4 cycle. So, this cycle is C4 cycle or Hatch and Slack cycle. Now, the product of photosynthesis, sugar, it requires carbon dioxide and water, carbohydrate, water and O2, which I have already discussed. Now, after C3 and C4 cycle, there is another process involved in the photosynthesis, which depends upon the light intensity, when the light intensity are present or not. That process is called photorespiration. As we have discussed earlier, during the photosynthesis, the carbon dioxide, it combined with water, it form CH2O, that is carbohydrate, and O2 and water. Like carbon dioxide plus water, it converted into C6H12O6 plus H2O plus O2. When we balanced it, 6. When we divide by 6, so this is CH2O, this is H2O, this is O2, here CO2 plus twice H2O. So, here is the carbon dioxide, this one, 2 H2O, 1 H, 2 H2O is here. Now, the product is CH2O, this one, O2, this one, H2O, this one. So, entire reaction we can summarize in this way. In dark reaction and light reaction, the splitting of water molecule into oxygen and electron, it takes place in light reaction, and this carbon dioxide is fixed into carbohydrate in the dark reaction. Now, this entire process which require chlorophyll, which is called raw material of photosynthesis, that is chlorophyll, light, these are the essential. Now, the rate of photosynthesis, it depend upon the light intensity. When the light intensity, as we have discussed in action spectrum and absorption spectrum, when the light intensity is high, the product is the O2 release is high. When the light intensity is low, so light intensity depend upon the rate of photosynthesis. During the photorespiration, what happen? During the daytime, from morning to midday, during midday the light intensity is high. During this hour, the RUBP we have already discussed, abundant enzyme Rubisco, abundant on earth. There is dual nature of RUBP. At high light intensity, rate of photosynthesis is high, oxygen concentration in environment during the midday is high, because photosynthesis so the concentration of oxygen during midday 12 hour to 2 p.m. there is a high concentration of oxygen present in atmosphere in comparison to the carbon dioxide concentration. So, what happened in these cases? The RUBP, the enzyme that is called Rubisco, it does not work with carbon dioxide, and Rubisco combined with oxygen. When RUBP combined with carbon dioxide, it forms phosphoglycic acid, and which finally converted into sucrose molecule, or which are the main product of the photosynthesis. But during this time Rubisco have double role, it does not accept carbon dioxide and it is going to be the combined with oxygen. So, the name has been changed RUBP oxygenase. So, in this case what happened? The RUBP combined with oxygen, it for 2-phosphoglycolic acid and 3-PGA. Now, the freshly formed 2-phosphoglycolic acid, it becomes activated and it forms glycolic acid during the midday. This entire process it takes place in chloroplast. Chloroplast we have already discussed, double membrane bounded structure, this one chloroplast. Now, this glycolic acid, the freshly formed glycolic acid during the midday, now it enter into the single membrane bounded structure that is called peroxisome. peroxisome, and here glycolic acid which transfer from the chloroplast, it converted into glycine. Now, this glycine transferred to the mitochondria. Mitochondria we have already known, and in cytology this is called powerhouse of the plant. This glycine now reaches to the mitochondria, and here it converted into serine. Now, this serine returned back to the peroxisome, and here the serine converted into hydroxypyruvic acid, and this hydroxypyruvic acid in peroxisome, it also converted into glyceric acid. Now, this glyceric acid reaches to the chloroplast and it forms 3 PGA, and in this process ATP is required, and this ATP converted into ADP, and 3 phosphoglyceric acid is formed. So, in case of photorespiration, 3 organelles are involved, which is called chloroplast, peroxisome and mitochondria. It is the net loss of plant, because the plant, the RUBP combined with the carbon dioxide, that form phosphoglyceric acid, which later on converted into sucrose molecule or starch molecule to fix the carbon dioxide, but here is the net loss of the plant, because the RUBP combined with oxygen. So, we can define the photorespiration, this is the inhibition, this is the inhibition of photosynthesis at high O2 concentration. This is also called Warburg effect. This Warburg effect or the photorespiration first studied by Decker and Teo in 1959. According to Krotkov 1963, he defined photorespiration is the extra input of O2 and extra release of CO2 by green plant. And the site involved we have already seen the chloroplast, peroxisome and the mitochondria, which involved in the photorespiration. Now, in the another chapter, there is the respiration. Here the term photorespiration and respiration is synonymous. And respiration. As we know, the respiration is a physiological process in which breakdown of carbohydrate in presence of enzyme or in absence of enzyme, it gives carbon dioxide, water and ATP. But in respiration there is no requirement of light, no light is needed. The photorespiration, as the term indicate, like respiration, but light is required. It occurs in presence of light. The second difference, respiration site is mitochondria, where ATP is formed, mitochondria as we can say cytoplasm also, because glycolysis takes place in cytoplasm and Krebs cycle takes place in mitochondria. But here 3 organelles are involved, that is chloroplast, peroxisome and mitochondria. The third one difference, it takes place in only in the green plant, but it takes place in all living cell. Here NADPH2 and ATP is lost. Here ATP and NADPH2 is formed, which is required for energy generation. Photorespiration is non-essential, we can say it is non-essential, but it is very essential. So, these are the differences between photorespiration and respiration. Respiration we will discuss in later on. Now, at the end of this chapter, one process is involved, that is CAM, which is called crassulacean acid metabolism. This plant is called crassulacean or CAM, crassulacean acid metabolism. As the term indicate, this process associated with the photosynthesis, it takes place in plant, the family is Crassulaceae. Example of Crassulaceae family: Crassula, Kalanchoe, Sedum. Beside the Crassulaceae family, it also occurs in the other plant like pineapple, Euphorbia, as we have already discussed in Euphorbia maculata, Euphorbia genus, both C3 and C4 are present, the euphorbia, agave, etc. The crassulacean acid metabolism, it takes place in hot and dry condition, where the condition are very hot, it takes place in, but in these cases the stomata of that particular plant close during the day hour and open during the night hour. So, all these processes are involved in the photosynthesis, which are discussed here, like light reaction, dark reaction, C3 cycle, C4 cycle, crassulacean acid metabolism. Thank you.